Good morning. How y'all doing? Look, the next thing we need to start looking at in this biology course is the role of DNA and its not only structure but its function. And I figure one of the best ways to start off is to go over what is known as the central dogma of biology. And I've got a nice little picture here. You've got DNA located in the nucleus of a cell. It gets transcripted to RNA, which is another nucleic acid, slightly different because of the sugar molecule, which then gets translated in the cell to produce the protein. And they got a nice little picture here. The DNA up in here, the transcription to make a copy of the DNA in an RNA molecule. The RNA molecule leaves, finds the ribosome, and its code is then turned into protein. It's a protein right on top there. There's a protein, and these three prongs read the three prongs in groups of three to bring the next amino acid into the chain of protein. Anyway, that right there is just a central dogma of biology. Feel free to Google it. Take a look at it. Hold on. Now, no discussion can really get going until we understand, probably at the base point, the fundamental structure of the DNA molecule. Up here I've got a pretty nice drawing, albeit it's schematic in nature, showing the double helix and its component parts. You've got the two sides, there's one side, and the other side of the DNA molecule. And each DNA molecule has this repeating unit right here. Three parts. There's a phosphate and a sugar on the outside that makes up part of the rail, the outside rail, and it stays consistent. Phosphate, sugar, phosphate, sugar, phosphate, sugar. And on the other side, phosphate, sugar, phosphate, sugar, phosphate, sugar. Now, notice that the sugar is a pentagon shape. There are 5 bonding sites for the sugar. And you start by counting 1, 2, 3, 4, 5. So on this side, it connects it. When I say it, I mean the nitrogen base, which is in here, and a nitrogen base, and a nitrogen base, and a nitrogen base. And on this side, it turns out to be, I think they call it the 3 side, but be that as it may, This is the component part, known as a nucleotide, right there. It consists of 3 parts, phosphate, sugar, and the nitrogen base. Notice that the only variety that occurs in a DNA molecule turns out to be the nitrogen bases. Now, you're lucky there's only 4 of them. There's only 4 nitrogen bases to be concerned about. An A and a T, a G and a C. And the A stands for adenine, thymine, guanine, cytosine. These are molecules. They are nitrogen, have nitrogen as part, atoms in them. And they make up the variety in a DNA molecule. And as it turns out, you will find that it's this variety that codes for the proteins, ultimately for the proteins. Another important part in this discussion is what is known as the complementary base pairing rule, also known as Chargaff's law, and that says when there's an adenine, it will bond with the thymines. When there's a guanine, it'll bond with the cytosines. So you can see, here's a thymine. What does it bond with? It bonds with an adenine. And if there were a cytosine on one side, it's got to bond with a guanine. And it's the sequencing here of these nitrogen bases that is the language, or the letters, like the letters to the sentence, that make up the proteins. One of the biggest processes that occurs with the DNA is called replication. The reason for the replication is to make a copy of the chromosomes in the cell. As the cell grows to a point, it will begin to undergo the processes of division. As you know, you started out as one cell, we'll call it the zygote. Before too long, you became two cells. Well, the DNA package that you had in that very first cell, which was a donation from mother and father, the two cells that are now going to come, each of them needs a DNA package, a complete set. So what happens, a process called replication occurs where this is making the copy of the DNA before the split. The process of the DNA replication, it begins when enzymes pull apart or unzip the helix. As you can see, it's called helicase. That's an enzyme. It's actually unzipping. As the two strands separate, the bases are exposed. As you can see, like that adenine has now got no complementary partner because it's been unzipped. Cytosine, thymine, it gets unzipped and exposed. The exposed bases then attract their complementary bases. So they're not exposed for too long before another enzyme will come along and will bring a nucleotide that has a nitrogen base that is complementary. As you can see, it matches right on up. And you can see from the top. The enzyme is called the DNA polymerase. And there we got DNA polymerase up there. And this replication process makes from one DNA strand into two identical DNA strands as the helicase unzips the entire 6 foot or so length. DNA replication occurs during the S phase of the cell cycle, and we did the cell cycle. And after replication has taken place, the chromosomes, as you've seen in many pictures, it shortens, and the shortening allows for a clean separation so that there is a new and complete package in each of the new cells. And anyway, that's that DNA replication. Very, very important. Each one of your trillions of cells is going to need that complete genetic package. So that means that you've got to copy your DNA package over and over and over at every time a cell splits so that the new daughter cells end up with the complete DNA package of the species. This occurs in plants, in protozoa, in animals, be they starfish, snails, humans, horses, mice. If you're a living organism, generally speaking, you've got DNA, and a multicellular living organism, each cell is going to need that package of DNA material. So you got to make a copy before you split. I mean, that's the long and short of it.